Interstellar everyone, welcome to class 12 biology. In this video we will discuss about the genetic code. In our previous video we discussed about the central dogma of molecular biology. The central dogma of molecular biology, which was proposed by Francis Crick, it describes the flow of information, the genetic information from DNA to RNA and from RNA to protein. So in our previous video, we discussed about the transcription process, right, in which an RNA was copied from DNA. So the next process will be the transfer of information from RNA to protein. So protein will be produced based upon RNA, the information contained within the RNA. So that particular process will be called as translation. But before we move forward with the process of translation, we need to understand how the information is stored within these genetic materials. And that information, the way in which the informations are stored will be called as the genetic code, right? How proteins will be produced from DNA or RNA, and how those informations are stored within DNA and RNA are called as genetic code. So if you look at the first two processes over here, replication and transcription, we have already discussed about replication and transcription. And in both these processes, we have seen the information is simple, right? Nucleic acid is produced based upon other nucleic acid. DNA is a nucleic acid, RNA is a nucleic acid. So in the replication process, a new DNA molecule is produced based upon an old DNA molecule, right? So that is information passed from nucleic acid to nucleic acid. And if you look at the transcription process, that is also the same thing, nucleic acid to nucleic acid, because both the DNA and RNA are nucleic acids. Now, but look, if you look at the translation process, what happens is a nucleic acid is directing the production of a protein molecule. So a protein as a biological macromolecule, it contains a long chain of amino acids, right? So there is no complementarity between a nucleic acid and a protein, right? A nucleic acid is a long chain of nucleotides, whereas protein is a long chain of amino acids. So nucleotides and amino acids, they don't have any complementarity. If you look at replication and transcription, you can see the complementarity, right? DNA is produced upon another DNA based upon the base complementarity, right? If there is adenine, a thymine will be added. If there is a guanine, a cytosine will be added. So there is a base complementarity, and same goes for RNA as well, production of RNA based upon DNA in the transcription process. But in translation process, when a protein is synthesized based upon RNA, there is no complementarity. So how the information is stored within DNA and RNA was a very complicated process, and it challenged the scientists during that time. However, scientists, they knew that any kind of changes, if any kind of changes happens within DNA and RNA during mutations, right? If there is any kind of changes in DNA and RNA, what happens is there is changes in the protein structure because there is changes in the amino acid sequence, right? If you make some changes in the DNA, what will happen? The protein will change because the information will change over here, right? If you make any changes in the RNA, again, the protein will change. And this change will happen by changes in amino acid sequence, right? So they already knew that the DNA and RNA are the genetic materials, but how these genetic materials store information for the production of a protein was a mystery, right? It was a big challenge. So genetic code, we can say that code that directs the synthesis of a chain of amino acids. So to decipher the genetic code or to figure out the genetic code, scientists from various disciplines like physicists, chemist, biochemist, geneticist, organic chemist, many of these scientists, they came together, right? And they tried to figure out the genetic code. So among them, a physicist called George Gamow, he came forward and he argued that since there are 4 bases in the genetic material, let it be DNA or RNA, if the genetic material is DNA, we have got again 4 bases, adenine, guanine, thymine, and cytosine. If the genetic material is RNA, we have got 4 bases, adenine, guanine, uracil, and cytosine, right? So since there are 4 bases and since there are 20 amino acids present within the proteins in biological systems. Therefore, he argued, codes should be made of combination of 3 nucleotides. Right? The codes should be made of combination of 3 nucleotides. So he was the first person who suggested that the codon is a triplet. Right? To prove that codon was in fact a triplet, as suggested by George Gamow, many scientists, they tried to decipher the genetic code. Right? So these are the scientists here: Hargobind Khurana, Marshall Nirenberg, Severo Ochoa. All of them are Nobel laureates. So they came together and provided proof of the codon being a triplet. Right? So let's look at their work, what they did. What Marshall Nirenberg did was, he developed a cell-free system for protein synthesis. What it means is, he developed a way in which he can synthesize a biological protein outside the cell. Right? So he developed a cell-free system of protein synthesis. And in his experiment, what he did was, he produced a synthetic RNA which contained a chain of uracil. So it is a polyuracil RNA. So in this polyuracil RNA was synthesized, and he put that in the cell-free system for protein synthesis, and he observed that this particular RNA, synthetic RNA, produced a protein which contained only phenylalanine as amino acids. So a long chain of phenylalanine was produced. And he concluded that the amino acid phenylalanine must be coded by a codon which contains a repetitive sequence of uracil. So the codon UUU codes for phenylalanine. In continuation, what scientists in his lab did was, they produced an enzyme, a Severo Ochoa enzyme, or polynucleotide phosphorylase, which helped in the synthesis of RNA with defined sequences. So they synthesized different kind of RNA over here, which contained now adenine only. So it is a polyadenine RNA. So a polyadenine RNA was produced, and then that was allowed to be translated into protein, and they observed that that protein contained only lysine, and therefore they concluded that for the lysine amino acid, the codon must be AAA. And same goes with a polycytosine RNA, and that produced a protein which contained proline as amino acids. So for amino acid proline, the codon must be CCC. And the rest of the codons were deciphered by Hargobind Khurana. So all 3 of them, they were responsible for deciphering the codons, right? So he produced RNA molecules with defined base combinations like U, C, U, C, U, C, U. So there is only U and C, right? U, C, U, C, U, C. So that is an RNA sequence over here. And he observed that this particular RNA produced alternating serine and leucine, right? Serine and leucine amino acid containing proteins, right? So that is how these scientists were able to decipher the genetic code. And now we have this particular chart. So this is the chart for genetic codes over here. We have got 4 bases, right? Uracil, cytosine, adenine, and guanine. So this is a basis present in RNA. So since the previous scientists, they worked on RNA, so here they have got 4 bases here, 4 bases here, and 4 bases over here, and they created these codons, which contains a combination of 3 of these bases, right? So we have got UUU, UUC, UUA, UUG, and likewise, right? So we have got a combination of permutation and combination of these 4 bases in triplets, right? So we have 64. We have got 64 codons in total, right? We have got 64 codons in total, and out of these 64 codons, these previous scientists over here, they figured out that 61 out of these 64 codes for amino acids, right? And 3 codons over here, I have highlighted them in red. 3 codons, they do not code for any amino acids, right? 3 codons, they doesn't code for any amino acids, and therefore they are called as stop codons. Okay, so this chart over here, this shows all the codons that are possible with these 4 nitrogenous bases present within RNA, and each out of these 64 codons, to 61 codes for amino acids and 3 doesn't code for any amino acid, therefore they are called as stop codons. Now let us try to understand the salient features of genetic code. The first one is the codon is triplet. So it is a simple thing, we already discussed about that. The codon is a triplet, which means a codon is made up of combination of 3 nucleotides, right? So there are 64 codons in total. Out of those 64, 61 codons code for amino acids, and 3 codons, they do not code for any amino acids, and they are called as stop codons. Second one is codon is read in contiguous fashion. There are no punctuations between each codon. Let us try to understand this one. In an mRNA, the codon is read in a contiguous fashion. If this is an mRNA over here, and we can see these codons over here in sequence, there are no punctuations, there are no gaps between the codons. Right? There are no gaps between the codons, there are no punctuations between the codons. The codons, they are read in contiguous fashion, like this, AUG, UUU, like that. There are no gaps in between. Okay, so this is what the second salient feature states. There is no gap, or there is no punctuation between each codon. Third is the codon is non-ambiguous. Code is a non-ambiguous. What does it mean? Each codon will code for a specific amino acid. Each codon will code for a specific amino acid. Let's revisit this particular table over here. UUU is a codon. It codes for a specific amino acid, that is phenylalanine. If you look at UAU, it will code for a specific amino acid, that is tyrosine. Right? UUU, it codes for phenylalanine. If you look at UAU, let's try to find UAU, it codes for tyrosine. Right? So each codon will code for a specific amino acid, and therefore a code is said to be non-ambiguous. There is no ambiguity. Right? UUU will not code for any other amino acid. UUU will always code for phenylalanine. UAU will always code for tyrosine. That's what the third salient feature says. Each codon will code for a specific amino acid. So that is the third salient feature. So the fourth salient feature is code is degenerate. Genetic code is degenerate. So these words are important that I have highlighted in blue, right? So code is degenerate. What does this particular term mean? It means one amino acid is coded by more than one codon. One amino acid can be coded by more than one codon. So let's look at the table again over here. This table is really important. You don't have to memorize, it will be provided to you during the exams as well. If you look in this particular table, let us try to understand why code is said to be degenerate. So I have highlighted serine over here. So amino acid serine. This is one amino acid. Serine can be coded by more than one codon. So we have got four codons which code for amino acid serine. UCU codes for serine, UCC, UCA, UCG, all these codons, they code for serine. So one amino acid can be coded by more than one codon. Therefore, the code is said to be degenerate. Genetic code is said to be degenerate. And same goes with proline as well. Right? Proline is coded by more than one codon. So that is what degenerate means. right? Code is degenerate. I hope you understood that. The fifth one, code is nearly universal. The code is almost universal. What it means is from bacteria to human, right? A codon UUU would code for phenylalanine, whether it be it in a bacteria. In the bacteria, if you have this particular codon UUU, it will code for phenylalanine. If this same UUU codon is present within our body, this UU will code for phenylalanine. So from bacteria to human, code is universal, right? UU will always code for phenylalanine, and the same goes with other codons as well. So it is said nearly universal because there are some exceptions as well, right? So the sixth salient feature is about this particular codon, AUG, one specific codon, AUG. AUG has got dual function. AUG is said to have dual function. AUG, it codes for methionine. It codes for amino acid methionine. AUG codes for amino acid methionine. And not just that, it also acts as initiator codon. AUG has dual function. It codes for amino acid methionine, and it also acts as initiator codon. In most of the mRNAs, messenger RNAs, AUG will be the initiator codon. AUG will be the first codon. So every amino acid sequence, polypeptide, the first amino acid will be methionine because AUG is the initiator codon. And the final one, the seventh salient feature will be the stop terminator codons, the stop codons. These three codons over here, UAA, UAG, UGA, these stop terminator codons, they do not code for any amino acid. Therefore, they are called as nonsense codons as well. Right? So these are the salient features of genetic code. So now let us try to predict the amino acid sequence produced by this mRNA sequence. So we have an mRNA sequence over here with these codons AUG, UUU, UUC, UUC, and so on. Right? So we have this mRNA sequence. Now by looking at this particular checkered board or this particular table, right, of genetic codes, can we predict, right, accurately, can we predict the sequence of amino acids over here? The answer is yes, we can. Right? AUG, it codes for methionine, right? It codes for methionine. UUU, it codes for phenylalanine. UUC again codes for phenylalanine, and so on. Right? So we can, just by looking at a sequence of codons, we will be able to form a sequence of amino acids coded by them. This shows that genetic code is non-ambiguous. There is no ambiguity that AUG will code for any other amino acid apart from methionine. There is no ambiguity that UU will code for any other amino acid other than phenylalanine. Right? So this is non-ambiguous nature of the genetic code. But can we do the reverse? Just by looking at the sequence of amino acids, can we accurately predict the sequence of codons? We cannot. Maybe for methionine, yes, because there is only AUG for methionine. But for phenylalanine, we have got two possible codons. Either it can be UUU or it can be UUC. So therefore, we cannot be sure which codon will come over here. So that is the second property of the genetic code. That is, it is what? Degenerate. Genetic codes are degenerate because one amino acid can be coded by more than one codon, right? So these are the properties, so we can practice like that. Now it is important to know that the codons are located on mRNA, right? So here we have an mRNA on which there are two codons over here, AGU and UAC. So the codons are located on messenger RNA, and messenger RNA was produced through transcription process by looking at DNA, right? So the codons are located on mRNA, and we have got these two codons, and this particular codon AGU codes for amino acid serine. This particular codon UAC, it codes for amino acid tyrosine. Now, who reads this particular codon? who is responsible for binding the amino acid in this particular sequence, right, by reading the codon. So who reads the codon? It is not the amino acid themselves that read the codon. Amino acids, they don't have structural speciality to read the codons, right? It needs the help. Between codon and the amino acid, between AGU and serine, there should be one adapter molecule. And that adapter molecule is nothing but transfer RNA, tRNA. tRNA acts as the adapter molecule between the codon and the amino acid which it codes for, right? So tRNA is said to be an adapter molecule because it has got 2 points, right? At one point over here, over the loop over here, it has got anticodon. Anticodon, as the name suggests, it is opposite of codon. It's not actually opposite, but it's a complementary base sequence of the codon. So the codon is AGU, the complementary will be UCA, and this particular complementary base sequence site is called as anticodon on tRNA. It has got one site which is anticodon. Anticodon will come and bind with the codon which is present on mRNA. And there is another point at the 3' end, there is the amino acid binding site. So this is the amino acid acceptor end. Right, this is the anticodon end, and this is the amino acid acceptor end. So at the 3' end, the tRNA, it is bound with serine, right? Serine amino acid, however you pronounce it. So tRNA, it acts as adapter between the codon and the amino acid. So same over here as well. The codon UAC, which is present on mRNA, it codes for the amino acid tyrosine, right? So between tyrosine and this codon, there is a tRNA which has got AUG as anticodon, and at the 3' end it brings tyrosine. So the amino acceptor site is tyrosine. So this is how the biological mechanism reads the codon present on mRNA. So the tRNA is the one responsible for reading the codon through its anticodon and brings the specific amino acids. So I hope it is clear, right? So the role of transfer RNA. So as the name itself suggests, transfer, it brings the specific amino acid on the codon. So now you should not confuse between codon and anticodon. If the codon is AGU, anticodon will be the complementary base sequence over here. And since this tRNA is also an RNA, you can see that A will pair with U, G will pair with C, U will pair with A. And same goes over here. U will pair with A will pair with U, and C will pair with G, right? And this transfer RNA, tRNA, they are very specific, right? They bind specifically with specific amino acids. And by that I mean the tRNA which has got anticodon UCA will bind with serine. The tRNA which has got anticodon AUG will bind specifically with tyrosine. So that is the specificity between the tRNA and the amino acids. So I hope how these codons are read and how tRNA is responsible for bringing in the particular amino acid which is coded by these codons. And you should remember that codons are present on mRNA, anticodons are present on tRNA. So in the final part of this video, we will discuss about mutations and genetic code. We have studied about genetic code, and they are present on either DNA or RNA, right? So how the alterations of the genetic code will lead to mutations. So we will try to understand that, and specifically we will try to understand the effects of point mutation. So insertion or deletion are two forms of point mutation. Insertion of one base pair, insertion or deletion of one base pair on the DNA will lead to point mutation, right? And then there is another form of point mutation called substitution as well, the effect of which we have studied in our last chapter, the example sickle cell anemia. Sickle cell anemia is caused due to a point mutation called substitution, where one base is substituted by another base, which leads to changes in one amino acid, and that change in one amino acid in the protein leads to formation of sickle cell erythrocytes, right? So sickle cell anemia was caused by a point mutation. Over here we will discuss about another forms of point mutation called insertion or deletion. Insertion or deletion of one base in the genetic sequence will lead to a mutation called as frame shift mutations. In order to understand what is frame shift mutations, an example is given in our textbook. This particular sentence is given: Ram has red cap. So just like a genetic code, it is composed of 3 letters, right? Just like codons, it is composed of 3 letters, right, each, and the meaning of which Ram has red cap, the meaning of each of these words leads to is equivalent to amino acid, a specific amino acid, right? Now what happens if we insert one letter before R, right? What happens is, now this will remain the same, Ram has will remain the same, but thereafter the codon has changed and the meaning has changed. That means the amino acid sequence has changed from this point onwards. Again, if we add a second letter over here, again the meaning or the amino acid sequence will remain changed from the point of insertion. So here you can see these two, when 1 or 2 letters are added, right, insertion of which means insertion of 1 or 2 bases changes the reading frame from the point of insertion. From the point of insertion, the entire reading frame changes. That means the entire sequence of amino acids will change. But if we add 3 letters, right, BIG between here, right, what happens? Ram has, it's the same like the original one over here, the same meaning, means the same amino acids, 2 amino acids, and now there is insertion of 1 amino acid, meaning because there is insertion of 1 codon, and the original red and cap amino acid will come back over here. So which means the insertion of 3 bases, if we add 3 bases, the 3 letters means 3 bases, the 3 bases, if they are inserted, what happens is it leads to change in or addition of 1 amino acid, 1 extra amino acid over here. However, the reading frame after that particular point of insertion will remain unaltered or unchanged from that point onwards. So that is what means by frame shift mutation. From here, these two represents the frame shift mutation. The entire reading frame changes if you add 1 or 2 bases in the genetic material or the DNA. If you add 1 base or 2 bases, what will happen? From that point onwards, from the point of insertion, the reading frame will change and the entire amino acid sequence will change, which will lead to mutations, and that particular mutation is called as frame shift mutations. Frame shift mutations are caused due to insertion or deletion. Insertion or deletion are types of point mutation, just like substitution, right? Insertion or deletion. Insertion or deletion of 1 or 2 bases will change the reading frame of the codons from the point of insertion. Let's take example of this same mRNA sequence that we have seen before, right? AUG, UUU, which goes for this particular sequence of amino acids. Now let's say there is a deletion of this particular U, which I have marked in red. Let's say this particular base is deleted or removed, right? There is a deletion, a point mutation happens, and this U is deleted. So what will happen? The base sequence or the codon sequence on this particular mRNA will change, right? It was earlier AUG, UUU, and UUC, but now look over here. After the U is deleted, what happens? AUG, UUU. Now what happens? Instead of UUC, we have UCU, right? And UCU over here. So after the point of deletion, the entire reading frame has changed. And because of this, what happens is the sequence of amino acid has changed. This is the effect of point mutation, right? So I hope you are clear about the point mutation and the frame shift mutation and how it leads to frame shift mutation, right? So in the next video, we will discuss about the translation process. right? How the mRNA will lead to formation of protein. So we will look at that particular process in detail. I hope you read about that one before you watch the next video. Thank you.